Hello, welcome back to Polymeric Biomaterials. We are now looking at the theme of biomedical polymers, and in the previous lecture we started looking at natural biomedical polymers, which we will continue in today's lecture as well. So, in the previous lecture we focused quite a bit on one very particularly important class of natural biomedical polymers, namely proteins. In this lecture we will focus on another very important class of natural biomedical polymers, namely polysaccharides. So, as part of today's lecture, we will first be looking at, as always, the structure of some of these polysaccharides, and we will also look at projections of these structures which are unique to these polysaccharides. We will recall some of our earlier discussions on isomer types, because as you will see, they will become important when we discuss polysaccharides. And then we will take three very important polysaccharides that are widely used, cellulose, chitosan and alginate. And as always, our goal here is to look at structure property relationships and how that affects function and performance. And towards the end of this lecture, we will quickly look at some other classes of natural biomedical polymers along the categories of Proteoglycans and Glycosaminoglycans, and then end with two examples. One is heparin, the other is hyaluronic acid. So, let us jump right into the lecture today, and we will first begin by looking at the monomeric unit in polysaccharides. So, the word polysaccharide itself suggests that it is a polymer, and so it is not very difficult to imagine that the monomer is called a monosaccharide. So, these are simply monomeric units that polymerize to form polysaccharides, and when I say polymerize, you have to recall that these would be covalent bonds. Now, typically these monosaccharides are short chains of covalently linked carbon atoms with typically hydrogen and oxygen hydroxyl groups attached to the carbons. And when we say short chains, we are typically talking anywhere from 3 carbons to about 6 carbons or so, triose to hexose. And for the purposes of this lecture, we will mostly be focusing on hexose, or the 6 carbon atoms that form these monosaccharides, because it turns out that those are the ones that will be useful for us to make these long chain polysaccharide molecules. And from a functionality perspective, and if you recall some of our earlier discussions, we have looked at the importance of the functional group. From a functionality perspective, there are two very important functional groups that are typically associated with these monosaccharides, and they are either the aldehyde group, in which case they are called the aldose, and one very famous example of that is glucose. And the other functional group, which we will not focus on much in this lecture, is but nevertheless important, is ketone, which becomes a ketose, and one very popular example of that is fructose. And note that both glucose and fructose, as we mentioned here, are 6 carbon atoms. Now, when we start to think about the structure of these monosaccharides, we will first look at the linear representation, or what is called the Fischer projection. And we are showing here glucose and two versions of it. One you see is called the D-glucose, the other you see is called the L-glucose. And what you will simply see is that one is a mirror image of another, and they are non-superimposable. Now, what distinguishes the D from the L is the placement of the hydroxyl group on the right side on the 5th carbon, or the farthest carbon from the functional group. So, the DL and L isomers are distinguished based on the placement of the hydroxyl group on the chiral carbon farthest from the functional group. So, the functional group here is this, and the farthest carbon from the functional group happens to be this carbon. This is the chiral carbon, and as we have already seen, the placement of groups on the chiral carbon make a difference between the isomers. So, the D isomer is one that is associated with the hydroxyl group on the right hand side, and the L isomer is associated with the hydroxyl group being placed on the other side. And it so happens that the D isomer is favored in nature. Now, a couple of points to note about these isomers. First thing is, they are mirror images, but they are non-superimposable, and typically the example given is those of your hands. They are mirror images of each other, but they are non-superimposable. We note that when we reflect the molecule on either side of the mirror, the whole molecule has to be reflected and not simply the fifth carbon alone. So, you see the entire molecule is reflected. Another important point to keep in mind is, while we are representing this way as the idealized structures, do remember that the bond angles are not 90 degrees. Just because they are shown this way does not mean that the bond angles are 90 degrees. This is just for our understanding and representation. The bond angles are actually the tetrahedral geometry associated with this kind of a structure, which we have seen earlier. Now, one can quickly imagine that when you have these 6 carbons based on 4 chiral centers, you can actually end up getting 2 to the 4 or 16 aldohexose sugars. This is the aldehyde group, and this is the hexose, which is the 6-member carbon unit. And out of the 16, 8 will be the D isomers and 8 will be the L isomers. And shown here are simply 2 examples of that, D mannose and D guelose. And when you place them right next to each other, you can see 2 things. One is they are both the D isomer. So, you see that the hydroxyl group on the carbon farthest from the functional group is placed on the right hand side, and you can also see that they differ in the placement of the hydroxyl groups in the other carbons as well, which makes them different not just in structure but in function as well. These two are 2 very different molecules, just based on the fact that they are both C6H12O6. But what you will see here, when you draw them out structurally this way in the linear Fischer projection, you can readily see that they are 2 very different molecules. And remember, here we are talking about configuration when we talk about isomers. So, neither are these mirror images, as you can see, nor can these bonds be rotated around like you do with conformations in proteins leading going from one to the other. You can't do that here because this has to do with the configuration of the molecule, which can only be changed when you break and form new bonds, which can't be done here. So, that should also tell you that although both of them can be represented as C6H12O6, they are both very, very different molecules, very different structures with very different functions. And there is a very popular mnemonic to remember these 8 or 16 aldohexose sugars, which a lot of students will be familiar with. All altruists gladly make gum in gallon tanks. Now, that is one way to remember the sugar names, but what I will emphasize here is, while the sugar names are also important, what is more important is to understand the principles guiding the structure rather than remembering the names. Now, there is a very interesting anecdote that Richard Feynman gives from his childhood. He talks about his father taking him on long walks and showing him birds, and he talks about his classmates who knew the names of those birds in different languages. So, he says, my friend could reel off the name of a particular bird in Chinese, in English, in Spanish and so on, and yet that did not mean that he knew anything about the bird itself. So, knowing the names of the bird was different from knowing something about the bird. So, that is a good analogy here for us to think about. Knowing the names of these sugars does not mean much. what's important here is to understand the following concepts: the idea of chirality, the idea of isomers, and the idea of the functional group. If you know these things, then you can always look up the names of these sugars in a textbook. So, but that being said, we will now move on to just quickly recapping some aspects of isomers that we have seen earlier. One form of isomers are the constitutional isomers, which are also sometimes called structural isomers. Where the two molecules will have the same molecular formula, but will have very different molecular structures. And shown here are two examples. One is glucose, which is an aldose, and one is a fructose, which is a ketose. And what you will readily appreciate is this one has an aldehyde functional group, this one has a ketone functional group. Another way to think about isomers is to look at stereoisomers, which have the same molecular formula and structure, but differ in the configuration, as we just said a while ago, which has to do with the way the atoms are oriented in three-dimensional space. Now, recall that this is not a new concept. We have seen this in very great detail in lecture 7. So, I am just going to recapitulate some key aspects of it as it pertains to polysaccharides and sugars. But if you are not familiar with these concepts, I would suggest that you go back and look up lecture 7 again to refresh your ideas on isomers, chiral carbons and configuration. So, under stereoisomers, you can have at least two types. One is enantiomers and the other is diastereoisomers. Enantiomers, like we just saw, are mirror images that are non-superimposable. So, D-glucose and L-glucose, where the whole molecule again is reflected, would be what you would call enantiomers. Diastereoisomers are stereoisomers that are not mirror images of each other. So, the difference could be occurring only perhaps in a few chiral carbons. So, for example, here you see D-glucose and D-mannose, and you see here that the difference occurs only in this chiral carbon and not the others. So, this is what you would call a diastereoisomer, which are not mirror images and of course not superimposable. Now, there is another very special category of isomers called anomeric isomers or anomers, where what the anomer means will not become clear when you draw the structure this way. These are essentially isomers that differ from each other in their configuration about the carbon 1 in the ring form. So, what we are drawing here is what you would call the Fischer or the linear form. What are anomers will become clear on the next slide when we start looking at how some of these linear structures can form cyclic structures through internal bond formation. And of course, just to drive home the point that these are all molecules that can be represented as C6H12O6, but remember that constitutional isomers are different from enantiomers, are different from diastereoisomers, and all the molecules shown on this slide are very, very different molecules. They have different properties. So, with that we now segue right into looking at understanding anomers in more detail. So, now we are going to look at the Haworth projection and try to understand what anomer means from the perspective of internal bond formation. So, in the linear structure we saw that you can have a functional group, and remember functional groups are reactive, that is why they are called functional groups. So, in this case there is a bond formation that occurs leading to intramolecular cyclization, where for example the C5OH group, hydroxyl group, reacts with the C1 aldehyde group, leading to what you would call a hemiacetal carbon, which is simply a carbon that is linked to OR on one side and OH on the other side. That is what a hemiacetal carbon is. And due to this new ring formation, there is a new OH group that is formed, and the carbon that was originally the aldehyde, that is the C1 carbon, which was originally the aldehyde in the linear form that we saw earlier, now that carbon becomes a new chiral center in the ring form, and that is called the anomeric carbon. So I've mentioned a couple of different words here. One is a hemiacetal carbon, which is simply a carbon that's linked to OR on one side and OH on the other side, as shown here. And this carbon, which was originally an aldehyde on the previous slide, has now, because of internal bond formation and internal cyclization, has now become a new chiral center with a new hydroxyl group that sticks out. And it so happens that this chiral center can lead to two different isomers called anomers based on the placement of the hydroxyl group here. So this is called the Haworth projection or the cyclic representation of the glucose that we saw in the Fischer or the linear form on the previous slide. And monosaccharides typically exist in equilibrium between the linear and the cyclic forms, and in solution you will typically almost always get the cyclic form, which is more stable. Now we have also added two new prefixes here, namely alpha and beta, and that's to represent the placement of the hydroxyl group on the anomeric carbon, namely carbon 1. So this is C1, this is C2, C3, C4, C5 and C6, and likewise on this ring as well. And the ring structure is unique because now in 2 dimensions we are limited by how we can show the ring structure, but essentially the carbons shown in the ring at the bottom, you can imagine they are facing us, and the carbons shown on the top in the ring are facing away from us, and the H and OH stick out on the top and bottom of the ring. So you have to imagine this in 3 dimensions. And the chiral carbon, which is C1, leads to 2 anomers, namely the alpha form and the beta form, and depending on the placement of the hydroxyl, if it is below it is called alpha, if it is above it is called beta. So now we have seen glucose, which is just a 6-member hexose, and it has an aldehyde functional group. You add the D, which has to do with the placement of the hydroxyl on the 5th carbon atom, the carbon atom farthest, the chiral carbon farthest from the functional group, and that is still shown in blue here. And then you add alpha or beta depending on whether in the ring form the chiral carbon 1 has the hydroxyl above or below the plane. So minute points here to keep in mind, but very important because they all have implications for how they can further join together to form specific kinds of bonds. Now with that stage being set, now we can ask, how do these ring molecules or these monosaccharides attach covalently to form polysaccharides? And it so happens that the bond that they will form is called the glycosidic bond, where the OH on the anomeric carbon, that is the C1, which we're showing in red here, reacts with the OH of an alcohol. So these OH groups across monosaccharides can react to form disaccharides, trisaccharides and polysaccharides. One important distinction to note, which you're not sort of glossing over here, but I'll mention it, this is what you would call an O-glycosidic bond, as opposed to an N-glycosidic bond, which you will find in ribose attached to bases in DNA. So the ring here is called a pyranose, which is simply a ring where you have 5 carbons and an oxygen, but in DNA, which we are not going to look at in this lecture or in this course in detail, you have what is called a furanose, which is a ring with 4 carbons and an oxygen. And it so happens that the sugar, the furanose ring or the sugar in DNA links to nitrogen bases using the N-glycosidic bond. That's a small interesting aside, but important difference to note. And what we are talking about here is the O-glycosidic bond. So now these glycosidic covalent bonds in the backbone typically occur between the 1,3 or the 1,4, and when we say 1,3 or 1,4, we are referring to the carbon atoms that we have already numbered here. That's why the numbering is important. Numbering always happens clockwise. And when you have small number of units, such as what we saw for peptides. In the case of a small number of amino acids being joined together, you would call them a peptide. Equivalent here is an oligosaccharide or an oligomer, more generally for polymers, or a polysaccharide when you have very long chains where multiple monosaccharide units are connected together. And shown here are examples of a disaccharide, maltose, and a polysaccharide, cellulose. So, in maltose or in cellulose, what's important to note here is not just what the individual monosaccharide sugars are, it's also important for you to note what type of bonds are formed. So, here they are D-glucose molecules. That should be obvious just looking at the structure of both of these mers, linked by alpha 1,4 glycosidic bond. It's alpha because of how this bond is formed based on the alpha anomer of the glucose, where the hydroxyl group on the first carbon sticks below the plane, and it is an alpha 1,4 because the carbon 1 here covalently attaches to the carbon 4 here. So, this would be 1, 2, 3 and 4. This would be 1, 2, 3 and 4. So, it is a 1,4 alpha glycosidic bond. So, all those details are important when you talk about disaccharides or polysaccharides. And likewise for cellulose, you can see that when you have many D-glucose molecules now linked by the 1,4 glycosidic bond, with the only difference being that it is a beta version, it is a beta anomer. So, you see that is reflected in how the bond formation is shown here, and it is still a 1,4. So, alpha 1,4 in maltose, beta 1,4 in cellulose, and both of them are D-glucose. So, this should give you a good sense of how monosaccharides or sugars can link covalently through the O-glycosidic bond. That should also become very obvious looking at how the linking happens to form large, long essentially chains of polysaccharides. Now, what we will now do is to use this understanding to look at three very specific types of polysaccharides that are biomedically important. The first one is cellulose. Now, cellulose is a very popular polymer, biopolymer. It's the most abundant naturally found polymer because it's found literally everywhere. Wherever you have plants and trees, you have cellulose. It's found in plant walls, and it is consisting of a linear, unbranched, long chain of D-glucose units. So, it's a homopolymer, which means it has only one type of monomeric unit that keeps repeating in the sequence, and the linkages, as we saw just on the previous slide, are given by the beta 1,4 glycosidic bond, which results in a long straight chain with, and because of the beta 1,4 glycosidic bond, each successive unit of the sugar is rotated 180 degrees with respect to each other, as shown in this schematic. So, you see here these rings are shown sort of in a three-dimensional kind of a structure for you to better appreciate where the hydrogens and the hydroxyl groups stick out. And you can see that each successive ring is rotated 180 degrees with respect to the previous ring. And that is because of the nature of the bond that is formed between the two units. Now, this repeating structure, you already have heard words in the last minute or so that should bring back some ideas about crystallinity to you. You have heard things like repeating structure, you have heard things like long unbranched chains, you have heard structures that are ordered. So, all of this should suggest to you that cellulose is going to be highly crystalline, and your guess is right, they are indeed very highly crystalline. And one interesting aspect about cellulose is they also have a very strong and rigid structure because of extensive intra and intermolecular hydrogen bonding. So, between chains, inter and within a given chain, they have extensive hydrogen bonding, which makes them extremely rigid and strong, like so. So, the high crystallinity immediately is going to lead to other kinds of properties that you can derive from a very high crystalline polymer, namely they are mechanically robust, chemically resistant. They are going to be insoluble in water or solvents, and the downside of course is from a processability perspective, you have processability issues which is going to limit their clinical application. Now, can you recall another example that we have seen earlier where we faced a similar problem? If you have gone through the lectures carefully, you will be able to immediately recall that we saw a very similar problem with PTFE in an earlier lecture, where you had very similar kinds of structures, although that was a synthetic polymer, and that led to high crystallinity, extensive interchain interactions leading to processability issues. And there we saw that Teflon FEP was used, where occasionally you will have substituents along the chain where the groups that stick out are perfluoromethyl groups get substituted, which is going to interfere a little bit with the overall crystal structure and the tight packing of chains, thus lowering the crystallinity and the melting temperature, thus improving processability. Now, if you are able to recall that example, you will be very easily able to relate to the topic I am going to talk about next, which is one important variant of cellulose, namely HPMC or hydroxypropyl methyl cellulose. This is popularly called HPMC, where the idea compared to Teflon FEP is not very different. Although they are two very different polymers, very different applications, conceptually you can think about substituents, as we saw for Teflon FEP vis-a-vis PTFE. Now we are looking at HPMC vis-a-vis cellulose that lead to defects in crystal structure, leading to less tighter packing and thus improving processability. So, you see, it is these kinds of patterns that you want to learn across polymers and not get stuck with specific properties of polymers, which are also important when you are designing polymers for specific applications. But in a course like this, what is more important to take away is concepts and patterns that you can now draw parallels across polymers, because when you come across a new polymer, then perhaps a similar concept will be applicable there as well. So, that is what is happening here. Here in HPMC, the OH hydroxyl groups are partly substituted either by methoxy or hydroxypropyl groups, which disrupts cellulose's crystalline structure, not unlike Teflon FEP, where the perfluoromethyl group substituents disrupt the repeating regular structure of PTFE, and here it leads to extensive hydrogen bonding with water, which also makes HPMC water soluble at room temperature. Now, HPMC is also interesting for another reason. It undergoes thermal gelation, where if you heat it beyond a certain temperature, it swells and gels upon heating, and this is a reversible process. These are physical crosslinks that occur. So, the fact that it is reversible should immediately indicate to you that there are not permanent covalent bonds that are being formed, but physical crosslinks that can be reversed upon cooling. And the ratio of these substituents, methoxy and hydroxypropyl, directly influence the solubility, swelling and thermal gelation temperature. Now, HPMC is a very important polymer from a pharma perspective, because it is used very often to control the release in extended release tablets. And the way it works is, when you coat broadly APIs and pharmaceutical formulations with HPMC, and when it enters the, when it encounters water, for example, what will happen is this polymer will immediately start taking up water and swelling. So, it will form a gel layer around. Now, that gel layer is going to slow down or retard the diffusion of the drug from within the matrix, leading to extended release. So, you see how we have related structure of the polymer to a specific functionality, namely its ability to gel, to a function, namely its ability to slow down drug diffusion, to application or performance, which is in extended release. So, it is a beautiful connection that we have made going from structure all the way up to performance. It is also used as a thickener and stabilizer in both topical and ophthalmic applications. One very popular application of that is these hydrocortisone creams, where it helps with spreadability of these creams. So, it is added as an excipient. Now, what I would recommend is HPMC and in fact its derivatives more than HPMC itself are very popularly used in pharmaceutical applications to prepare what are called amorphous solid dispersions, and that is because HPMC can gel, it can be made water soluble, and it is highly processable, none of which was possible with cellulose. So, you see, by making some small changes in the structure, we have completely changed the properties to be able to make it useful and processable from an engineering or design perspective, making it processable for specific pharma applications such as in the preparation of amorphous solid dispersions through a very, very important process called spray drying. So, I would highly encourage you to see, look up and see how HPMC's derivatives are used particularly in spray dried formulations for preparing amorphous solid dispersions. So, now that is cellulose. We will next look at back to back two charged polysaccharides. One is a positively charged polycation, the other is a negatively charged polyanion. One is chitosan, the other is alginate, to further look at how charge plays an important role not just in its properties but also in its function and performance. So, we are moving to chitosan. Chitosan is a very, very famous polymer, again widely used in many applications both in research and in the clinic. It is an amino derivative of chitin, which is found in crustaceans. It is a linear homopolymer of N-acetyl-glucosamine, that is chitin, and chitin structure is shown here. So, you can see that the rings have these amine groups that stick out that have been now substituted with the acetyl group as shown here. So, that explains the name N-acetyl substituted glycosamine. So, that should explain the structure of chitin itself, and you see every single of them, every single member of the chain will have the N-acetyl substituted glycosamine in regular repeating units. Now, that is chitin. What you can do when you get chitin from these crustacean exoskeletons is to deacetylate with either sodium hydroxide or through enzymatic reactions such as chitin deacetylase, which is fairly intuitive. Remember, ASE ending is for enzymes, and this is literally going to deacetylate chitin, resulting in these primary amines now, but not everywhere, on some but not all. So, you see some of it still retains this N-acetyl substitution, some of them have been deacetylated, leading to what you would call chitosan. So, now chitosan can be thought of as a random copolymer depending on how the deacetylation is done and how much of it is done between D-glucosamine and N-acetyl D-glucosamine. So, all of this should make sense looking at the structure and how the deacetylation is done. Now, it turns out that this has some very interesting consequences for the properties of chitosan. Chitosan typically will have a fairly large percentage of deacetylation, typically more than 50%, and the molar mass can vary widely from say about 50 kilodaltons or so to greater than 300 kilodaltons. So, we are talking about long chains here, and this is what you would call a polycation, or more generally what you would call a polyelectrolyte, which is a polymer where you have the presence of a single type of charge along the chain. So, the surface charge that you get because of the primary amines, which can get protonated, is obviously proportional to the degree of deacetylation, because you see that cannot happen with this, can only happen with these kinds of groups. And this at body pH it is positively charged, which means it now has adherence ability to negatively charged tissues. such as mucosal surfaces. Sometimes these are also called mucoadhesive materials, where they can attach through electrostatic interactions to mucal surfaces, and that's as you can see, is a functionality which comes from the property of charge, which in turn comes from the presence of the charge due to the deacetylation. So again, this is backtracking from function to property to structure. Chitosan can undergo gelation and through entangled networks and gel at either by increasing the pH or through polyion complexation. So you can have oppositely charged ions that can link multiple chitosan chains, that's polyion complexation. And chitosan can be metabolized by enzymes such as lysozyme, which makes it biodegradable. Now, the presence of the amine groups and multiple hydroxyl groups makes it very, very interesting from a design perspective. Now, you can functionalize chitosan further by using these functional groups as starting points for attaching other kinds of molecules. If you want to attach proteins, if you want to attach peptides, all of that can be done through some very simple click chemistry kind of approaches based on the presence of these kinds of functional groups that can further be modified. And one very important application of chitosan based materials is in hemostatic dressings. Hemcon, for example, was quite popularly employed in the Iraq war for stopping bleeding, and the ability of chitosan to stop bleeding and immediately clot and be used as a hemostatic dressing can be directly linked to charge. So that's the connection that you want to make. Likewise, we can now look at another polymer that's negatively charged, namely alginate, which is also a poly electrolyte but a polyanion. Alginate is a linear anionic polymer derived from brown algae. So you see, all of these are naturally derived polymers, cellulose from cell walls of trees, plants, etc. As we saw, exoskeletons of crustaceans for chitosan, and now brown algae, which is the source for alginate. And alginate has two different types of monomers that are present. One is the mannuronic acid, the other is the guluronic acid, which are usually abbreviated as the M and the G units, and it's a 1,4 linkage between a beta D mannuronic acid and an alpha L guluronic acid. Now, I hope that these distinctions are clear, what the L and the D mean and what the alpha and the beta mean. They are both important and the two are very different. So, I hope you can now go back and reconnect this to some of the concepts we saw at the beginning of this lecture when we looked at both L and D isomers as well as the alpha and beta anomers. Alginate can have blocks of either similar or alternating sections. So, you can have either MMM, GGG kind of sections or MGMG kind of sections. And like chitosan, alginates can also go from somewhere from like 50 kilodaltons to maybe greater than 500 kilodaltons. So, all of these are very long chain molecules. Now, just as we saw with chitosan, which can undergo polyion complexation, alginate can be cross-linked in the presence of divalent cations, particularly calcium. And shown here is a schematic of how this cross-linking can happen. Essentially, the primary functionality in alginate comes from the COO minus present in alginate, and you can imagine that two of these negatively charged COO minus on two different chains can now be cross-linked through one divalent calcium ion. So, you can have this is one chain, this is one chain. So, you can have electrostatic interactions or cross-linking, ionic cross-linking between CO minus here on one of the G units and CO minus here on another G unit in a different chain, and the two of them can be bound. Calcium is also interesting because it forms this egg box like structure as shown here. So, the red dots represent the calcium ion and the black wavy lines represent the chains, where the CO minus from two different chains can be now cross-linked through one divalent cation like calcium, and calcium is unique in that its size makes it suitable for these chains to form these kinds of egg box models, which some of the other divalent cations may find it hard to form because of different sizes. Calcium also has been postulated to more than bind just to the two CO minus groups. It can also coordinate between the oxygens on the hydroxyl groups across chains. So, there are some interesting theories out there for that kind of coordination that calcium can form between hydroxyl groups as well, in addition to the interactions shown here. Now, that leads to specific kinds of properties and functionalities. So, first of all, we will note that it is largely the G units that cross-link. So, the M to G ratio and the calcium to G ratio affects properties of the gel that forms, for example, stiffness, right. More cross-linked it is, stiffer the gel, but higher cross-linking will depend upon how much calcium you have and how many G units you have. And alginate is degradable, quote unquote, in the sense that the degradation occurs more by exchange, ion exchange in the presence of aqueous buffers such as PBS, where sodium can now slowly exchange with calcium, and because sodium is not divalent, they will lose this egg box structure model and the chains will just come apart. So, when we say biodegradation, what we actually mean is dissolution, where the chains just come apart. We are not specifically referring to chain scission here. We are talking about the cross-linking being undone. That is what we mean here. Alginate also is a very, very important polymer for applications that involve controlled studies in vitro, where people want to investigate interactions of specific cells to biomaterials under controlled environments. And that is because polymers like alginate and even polyethylene glycol, as we have seen earlier, are what you would call biological blank slates. So, intrinsically they do not have interactions one way or the other with biological systems because they lack recognizable sequences, but they can be very easily modified with ligands such as RGD. Now, this should recall something that we have seen in a previous lecture for single letter codes for amino acids. So, you can go back and check what R, G and D mean. Or functionalization with proteins such as fibronectin, which in turn cells can recognize and attach. So, polymers like alginate, for example, while they are widely used in drug delivery and in tissue engineering applications, and there is a lot of research out there that goes on with alginate. One very important application of alginate based materials is also to study cell responses or biological responses in controlled microenvironments through very, very controlled interrogation with specifically modified biomaterials. And one important product that is out there is this Phytocare hydrogel dressing that has received this 510K clearance, FDA clearance for partial to full thickness wounds. So, that is alginate. Now, we will look at one particular class of again charged polymers, natural polymers, but they have two different functionalities. These are proteoglycans, but they have both a protein group and a glycosaminoglycan group attached to it. So, these are biological molecules that have a core protein which are covalently attached to glycosaminoglycans or GAGs. And one example of that is shown here, which is Aggrecan. It is a very important and famous proteoglycan. What you see here, the full structure is the Aggrecan aggregate, which If you now zoom in, you can start to see the Aggrecan molecule itself, and already you can perhaps appreciate that we are talking about a hierarchical organization, which we have alluded to and actually looked at in great detail when we looked at collagen. So, you see the hierarchical organization of natural polymers or biologically derived polymers are a very interesting way and beautiful way to appreciate order and organization in polymers. So, what we are seeing here is that they have this bottle brush like structure as shown here, and they have very tissue specific functions. So, for example, small proteoglycans participate in collaboration with collagen for their fibril growth and organization. Because of the large presence of charge which largely comes from negatively charged GAGs or the glycosaminoglycans, they help absorb and retain water in tissues because of the polar nature, and cell membrane bound proteoglycans can bind collagen and ECM and other growth factors. So, they can serve as these repositories of growth factors and can release them at will depending on the need. And what you see here is an Aggrecan aggregate, like we have said, where there is a protein core and there are these chondroitin sulfate and keratin sulfate glycosaminoglycans that stick out. So, there is a protein core for Aggrecan like for any proteoglycan, and then there are these long chain glycosaminoglycans that stick out. So, it includes both a protein core and a polysaccharide set of chains which are largely negatively charged. Of course, here you also see another level to the hierarchical organization where all these are now non-covalently attached to the hyaluronic acid or hyaluronin chain. So, glycosaminoglycans which are now attached to proteoglycans which form the polysaccharide part of the proteoglycan, these are essentially long unbranched polysaccharide chains with a repeating disaccharide unit. Now, this disaccharide unit is typically hexosamine, either glucosamine or galactosamine, and a hexose or a hexuronic acid such as the galactose or guluronic acid or hydruronic acid, either or both sulfated, and that leads to a lot of negative charge. So, the sulfate and carboxylate groups of the sugar give rise to a very large negative charge density, which primarily helps in making the molecules polar, which help in absorbing water and retention of water, sequestering growth factors. All of this functionality comes from charge. And some examples of these are hyaluronic acid, chondroitin or dermatin sulfate, heparin sulfate, keratin sulfate, and we see here that both keratin sulfate and chondroitin sulfate are involved in Aggrecan. Hyaluronic acid is an exception because it is one of those only GAGs that is not part of a proteoglycan family, which we will see in just a minute. So, again here is an example of a set of molecules or a family of molecules where the hierarchical organization and the structure gives rise to specific kinds of properties, for example, charge density, which leads to some very important functionality. So, these are the points that you want to appreciate from a structure like this. Heparin, which is related, as an example, we are seeing also has very high negative charge due to sulfate and carboxyl groups. As we have seen for proteoglycans, especially the GAGs, these can now bind growth factors such as VEGF and fibroblastic growth factor and can regulate the release within the ECM or the extracellular matrix depending on the strength of the association. So, the extracellular matrix, which is the matrix surrounding mammalian cells onto which they deposit proteins and they are held together in the form of tissues because of the ECM. One of the major components of the ECM is collagen. Another very important component, which is a structural protein, another very important component are these proteoglycans, which now can help sequester things like growth factors based on charge and can help release depending on the need. And heparin is not a new molecule. We have looked at it in an earlier lecture when we have looked at antithrombogenic coatings. And so it is used very widely in the clinic as an injectable antithrombotic. And earlier we would have simply said that the charge leads to antithrombogenic effects. Here we are looking more at the mechanism for how that is achieved. So you have antithrombin here and you have heparin here, and heparin has this pentasaccharide sequence which can now bind to a positively charged region on the antithrombin, which is called the heparin binding domain as shown here. So this binding of heparin or the pentasaccharide sequence on heparin to the heparin binding domain, which is positively charged on the antithrombin, results in a conformational change. So this binding results in a conformational change in the antithrombin, and so thrombin, which is now active, once it binds to the conformationally changed form of the antithrombin to form the thrombin-antithrombin complex, the thrombin becomes inactivated, thus preventing thrombosis. So you see the sequence of steps that leads to this: the negatively charged pentasaccharide sequence binds to the positively charged heparin binding domain on antithrombin, and it's important that that binding happens because that binding leads to a conformational change in antithrombin, which is only when the antithrombin can sort of take in the thrombin and form the complex to inactivate it. So this is the mechanism broadly through which heparin exerts its antithrombotic effects, and that's very well utilized in this propatin graft, which are just thromboresistant bioactive surfaces made thromboactive or thromboresistant through heparin surface functionalization. So these are essentially EPTFE, expanded polytetrafluoroethylene grafts that are now heparinized or heparin functionalized to make them antithrombogenic, and they are used for bypassing blocked arteries in legs in peripheral artery disease or vascular access grafts as well. Now this is an interesting combination for two reasons. One is EPTFE is a synthetic polymer and heparin is a natural polymer. So you see a beautiful blend of a synthetic polymer with its own set of advantages combined with heparin, which is a natural polymer with its own set of advantages, to sort of leverage the best of both worlds to create a material that can have perhaps the mechanical properties and the durability of synthetic polymers such as EPTFE, but also have favorable interactions, not causing thrombogenesis etc. Through heparin functionalization. Another important aspect to note here is this largely contributes to bulk properties. This largely contributes to surface properties, both of which are equally important in biomaterial design. We have not looked at surface properties much in this course just yet. It's a topic we will look at in some detail later in the course, but it's something to keep in mind because, like you see right here, the EPTFE surface has to be functionalized, but heparin on its own will not make the cut either, because to function as vascular access grafts, you want something like EPTFE. So it leverages the bulk properties of a synthetic polymer along with the surface properties of a natural polymer. So it's a very nice, interesting combination of both types of polymers. The last polymer under this natural polymer set that we will look at is a polymer we have just alluded to, which is hyaluronic acid, which, like I said earlier, is one of the only GAGs that's not part of the proteoglycan family. It's non-sulfated and is not associated with a protein. Typically, under physiological conditions in the body, it occurs as the sodium salt. So it may also be called sodium hyaluronate, and it is characterized by an extremely large molar mass. So you can reach something like 5,000 kilodaltons perhaps, which is huge with hyaluronic acid. it has a very high water binding capacity like with all other GAGs. Interestingly, it also forms these lubricious solutions which now help hyaluronic acid be used as viscous supplementation in joints. So, this is one of the therapies that's initially recommended for patients suffering from osteoarthritis, where hyaluronic acid could be injected into the knee to bring back some of the viscous supplementation in the joint where the cartilage may have started to degrade. It's also used as an alternative to collagen for skin fillers in reconstruction, and the effects that it has will depend on the cross-linking water content and the particle size, which is not hard to imagine because again it's the structure that determines the properties that determines the functionality and performance. In closing, what I would like you to do is pause and look up this very important idea that integrates all the concepts we have seen so far under natural polymers, and that is the role of collagen, aggrecan and hyaluronic acid, all three of them. Collagen is a protein, aggrecan is a proteoglycan, hyaluronic acid is a glycosaminoglycan. Three very different classes of biomolecules or biopolymers. They have very distinct functionalities in the articular cartilage. Each of them contributes something specific to the way the articular cartilage performs in the knee, particularly in terms of its mechanical properties, both in tension, in compression, water binding capacity, all of this. So, you will see when you look this up that it's not one but the combination that actually provides the articular cartilage its functionality, and you will be able to better appreciate how the structure of each of these leads to its properties and its specific functionality within the articular cartilage. So, with that we will end this lecture on natural polymers.